Mr. Chairman, ladies and gentlemen, it's a pleasure to be here in a city where I used to work for a couple of years quite a while ago. I thought what I'll do today is to present to you not the details of the work in my lab, but rather give you an overview of the things that are going on in receptor research and that might be leading to the drugs of the future. So I'll be talking about principles that various researchers who've helped me in preparing this talk are actually working on in these days. Let me begin with the mechanisms of receptor activation, and I'll show here a few slides that I borrowed from Brian Kobilka. It turns out that activating a Gproteincoupled receptor is actually a very subtle event. What you see here is the receptor structure, so the helices, the amino acids, the side chains that bind the ligand, and depicted here in orange is the antagonist ligand carazolol. So this is a receptor in its inactive state. If we now superimpose the structure of the same receptor bound to epinephrine or adrenaline, the full agonist, in green, you see the active receptor structure. So again, the helices and the second ligand placed here. You'll see that it looks almost identical. The differences between the active and the inactive structures are really minute. At the point in the binding site where the distance is greatest, this is these two amino acid side chains, you have a difference of only 2 angstrom, which means that it's a very, very tiny movement that's induced by the agonist or is induced by drugs. And it also tells you that the room to play with, if you want to develop a receptor-directed ligand, at least in some instances, is very, very small. It appears that there is not much you can do between an active and an inactive structure, and so drug development at this site can be very difficult. In some other receptors, this movement is a little bigger, but in all cases, it's quite limited. So what can we do to develop new types of drugs? One example comes from an already existing drug, salmeterol, which is a beta-2 agonist, not quite full agonist, that's used in asthma therapy and is characterized by a very long half-life. It was originally thought that this drug, which has a long hydrophobic tail, binds somehow to the cell membrane, but what really happens is that this tail, this is the yellow compound here, that this tail extends to the outward face of the receptor into a second binding pocket, which is a binding pocket that occurs in a number of Gprotein-coupled receptors and that has recently gained quite a bit of interest. We call this binding pocket, into which all the adrenergic ligands bind, the orthosteric binding pocket. So this is where the endogenous compounds go. And this additional site that's utilized by salmeterol, we call the allosteric site. And for a number of receptors, compounds have been identified that bind exclusively to this upper allosteric site and influence the receptor in multiple ways that can be interesting to develop drugs. Arthur Christopoulos from Melbourne has modeled a number of receptors. So this is, to the left, this is the general model of these receptors, and he's modeled a number of receptors with their binding pockets. And this slide here just depicts the various binding pockets and their position in the receptor where they lie. And what you'll see here is that the binding pockets can be all over the place of this central core of this receptor. Some are very much to the extracellular phase, others are right in the center. Now, these are all different receptors, but here for the calcium sensing receptors, there are a number of compounds that actually bind to different pockets, and using those different pockets, we hope that we can one day influence the receptors in very specific ways. An example where this has been studied in some detail and where promise is at least almost imminent is the GLP-1 receptor. This is again from a paper of Arthur Christopoulos, where they try to develop the principles that we may utilize to develop new types of drugs. What you see here in red is the receptor structure. To the top you see, okay, with this weak red point, you see the extended N-terminus that characterizes these receptors, and the ligands, the endogenous ligands, bind to a pocket in the transmembrane region and also to the extended terminus. Now there is, in this schematic view, there is a number of ligands that bind to this orthosteric pocket, but in different modes. And so the hope is that with these different binding modes, one might be able to cause distinct downstream signals. So if you have, for example, two pathways here, term P1 for pathway 1 and pathway 2, the hope is that with one or the other of these orthosteric ligands, you can produce one or the other of the signals or have quantitative differences between the two signals that you are producing. In this receptor, there's also an allosteric site, so a site that's not touched by the endogenous ligands. And again, the hope is that by occupying this allosteric site, you may modulate the effects of the first orthosteric ligands. So by combining two ligands, you might switch a receptor into one or the other signaling pathway. In a very recent study, from Patrick Sexton's lab that's in Press in Cell, they've actually shown that this is true and that it can be utilized by various ligands. So what they've done in this study is they've mutated every single outside or surface accessible amino acid, so it's a painful piece of work. And by studying all these mutants of the receptor, they realized that different ligands bind to different sites in the receptor. The endogenous ligand, GLP-1, binds in blue here, okay, so it binds to this part of the outer surface. And oxyntomodulin, a ligand that is biased more towards the ERK pathway rather than to the G protein pathway, this second ligand binds slightly different. And the signal that comes from the binding is transmitted through this receptor molecule along different paths that are depicted here with these dotted lines. So the basic idea is that you can bind to a receptor on this side or on that side, and that transduces to the intracellular phase and ultimately results in the activation of one or the other pathway, allowing us, hopefully in the future, to develop drugs that will differ in their downstream and therefore also in their physiological effects. What we learned from these studies, and I'll just show you two slides about this, is that the transmission of the signal across the single receptor molecule is actually quite flexible, and that there are multiple pathways that can be utilized for receptor activation. In fact, it appears that the receptors are kind of floppy molecules. They don't switch like a light switch. They just move slowly, and when we determine the kinetics of the initial steps of ligand binding and ligand-induced activation of the receptor, and the more downstream effects that are seen by the cell from the inside, that are seen by the G protein, we realize that these first changes here occur very fast, on the order of 5 milliseconds or so, but the movements that occur at the intracellular phase of the receptor take much longer, 5 to 10 times longer. So the transmission of a signal across a receptor is slow. And that's been postulated already a few years ago by Brian Kobilka when he studied receptors with NMR structures, and he uses this type of cartoon to depict the receptor as a floppy structure. So even when the outside is quite fixed. So you see here the receptor with its binding site that can be occupied and thereby stabilized. You still have a lot of mobility on the intracellular phase, and the transmission of a signal from the outside to the inside of the receptor takes a while and is a not very reliable process. And the receptor is active only at the time when both an agonist is bound to the outside and a G protein or a similar protein is bound to the inside. Not only receptor protein is floppy, but the whole situation is floppy, as is depicted in this cartoon by Marta Filizzola, with whom we're working in an NIH project on opiate receptors. So she models these receptors, these very colorful receptors on the surface of a cell, and what this slide is supposed to show you is that these receptors are moving rapidly on the surface of a cell. They can aggregate and disassemble again, and this is a highly mobile film, cartoon. But the reality is as mobile as the cartoon is. This is a film that was done by Davide Calibiro in the lab, and you'll see here how individual receptors, TSH receptors, move on the surface of a cell, and you see that this is a rapid movement. And not only the receptors, but also the downstream proteins, the G proteins, move rapidly on the surface of a cell. Here you see red receptors and green G proteins as they move on the surface of a cell, and they do contact each other from time to time, but you see that the whole thing is quite a mess. Now, when you look at a film like this, you may have the impression that the receptors are evenly distributed over the surface of a cell. But this does not seem to be the case, and that's actually an issue that is fairly unknown, but maybe very important therapeutically. A nice example of such a heterogeneous distribution comes from the lab of Nigel Birdsall, who has studied in this example muscarinic receptors in the heart, in the mouse heart. Now, this is a really classic example where you might think that a tissue or in a tissue, each cell expresses these receptors and responds similarly. But if you look at these cardiac myocytes, in red, this is the surface of the cell, in the atrium of a mouse heart, you see that only very few here in green express the receptors, and others don't, for reasons that we don't understand at all. But it means tissues are not homogeneous, even if you think the tissues contain the same cells and the cells should be more or less equal. It seems that they're very different, and there may be responsive cells and there may be non-responsive. And the interesting question is, what are the differences between those cells? And does it occur only in the heart or also in other tissues? An example that we studied with Stefan Schulz in the lab already some years ago shows this for the pituitary, where we have a number of cells that contain a cyclic AMP indicator, so we can find out to which ligands they respond. And among cells that are supposed to be the same, you find some that are responsive to hormones or to synthetic ligands and others that are not, indicating that maybe in tissues we have cells that we think are the same, and yet they express different receptors and they have different responsiveness, which allows a lot of heterogeneity in responses and maybe also in utilizing therapeutic effects by combining drugs that can activate or inactivate specific cells. Another issue that has recently gained much interest is the fact that within a cell, the receptor signals are not homogeneous. The traditional view was that you have a cell that is like a ball, and wherever you stimulate it, you get an intracellular response, such as an increase in calcium or an increase in cyclic AMP, and that would be homogeneous and result in a response, like you see in a liver cell and its metabolic response. However, recently it became apparent that the situation is much more complex, and that even within a cell you can have heterogeneity of where the receptors are and what they do in a cell. And so the overall idea that we're trying to develop is that receptor signals in a temporally and also spatially defined pattern, and that drugs of the future will utilize only a certain spectrum of this pattern. In a cartoon that we draw here in a commentary to work by Mark von Sastro, we try to depict this in a schematic way, saying that initially receptors are activated at the cell surface at the top left and signal the traditional way as we know it from our textbooks, like lead to stimulation of cyclic AMP and produce a wave of cyclic AMP. In the second step, the receptors are then internalized and they produce another signal that is spatially distinct. So it occurs in these clathrin-coated pits or vesicles in the center, and it's biochemically different. In this case, it results not in activation of cyclic AMP, but in activation of ERK kinases, so protein phosphorylation signal. And a third wave, you see that receptors can then move from the cell surface to vesicles. Here is the receptor in a vesicle where they meet again with G proteins and produce a second wave of cyclic AMP. And this second wave of cyclic AMP may produce measurable cyclic AMP in the cell, but the cyclic AMP has a different flavor. It causes, at least in some instances, different physiological consequences. All these events can be recorded, for example, by putting fluorescent labels in these proteins and studying then such events in the microscope, as you can see in this film here. You can see the colors of these receptors changing, and so you can follow on the surface of a cell where and when these receptors are being activated. And you see it's a rapid process that occurs over a few hundred milliseconds in most instances, and you can see that it also happens in a localized and very specific manner. So it allows cells to mediate very different functions. Looking at the speed of this, we see something that I referred to already, that the initial steps are quite rapid. They occur on the initially probably on the 5 millisecond step, and then things get slower and slower as we are more downstream, and then the cellular response takes quite a while. But it's all very short-lived. The activation process of these receptors and the downstream signal takes about a second or so for most receptors that we are studying. And then the receptors become phosphorylated by GRKs, this well-studied process that leads to inactivation of the G protein-mediated signaling wave, but at the same time, it allows the receptors to interact with beta-arrestins here in blue and produce the second wave of signaling that's mediated by this. And if we look at the kinetics of these different signaling waves, for example, for the PTH, the parathyroid hormone receptors, we realize that the receptors are never statically signaling to one pathway, but they're producing one wave of signals and already start, while the first wave is going on, the second wave. Here you see occurring over a few seconds the stimulation of a receptor that leads initially to binding in green, so it's a quick binding process occurring over a few seconds. Then in red, you see the activation process, activating the receptor conformation and then activating the G protein, and you see that it happens very shortly afterwards. The dashed black line shows you the beta-arrestin binding, and you realize the beta-arrestin binding occurs almost as the receptor is still busy signaling. And it's only in this very brief period between activation and talking to the G protein and binding of the beta-arrestin that the receptor couples to a G protein. So we call them G protein coupled receptors, but only the very short interval in red is the short period of time in which the receptor is actually coupled to a G protein and signals. And then a second wave follows, which is the beta-arrestin mediated wave. Now the first process, the process depicted in red, takes a while to work its way downstream into the intracellular space and to cause accumulation of cyclic AMP, the classic signal. But by the time the cyclic AMP goes up, the receptor no longer stimulates the G protein, so it produces one wave after another. These waves are using modules that are very similar. The G protein module is something you've learned many times from textbooks. The receptor activates a cycle of activation and deactivation of the G protein, and this produces a cycle of activation and deactivation of the effector proteins. We now believe that the same is true for the activation and deactivation cycle of beta-arrestins. So beta-arrestins are inactive. They can assume an active conformation triggered by the interaction with the receptor. This active conformation then triggers a cycle of a downstream kinases that switches from inactive to active and ultimately produces an effect. So in these different signaling waves, there are modules that are being used time and again that are consisting of cycles of activation and deactivation, and this occurs in downstream steps as well. These were the temporal patterns of signaling, but there's also a difference in spatial patterns. So there's a cyclic AMP signal that's being produced at the cell surface and produces a first wave. There's a second wave of cyclic AMP that's produced from receptor signaling in intracellular sites where they're also coupled to G proteins. And it appears that the cell can distinguish between cyclic AMP that's generated here and that's generated here. How it does this is still a puzzle. I'll show you some possible answers to this. But it may have important functional consequences, as I'll try to explain to you in this complicated slide that looks at the response in the thyroid to TSH receptors. What you see here in these thyroid cells is actin fibers and phospho-VASP. It's the changes in the actin fibers and the phosphorylation of VASP that coincide with activation of these cells and ultimately hormone secretion. So here you see the effects of TSH, you see the actin cytoskeleton being remodeled, and you see the occurrence of phospho-VASP. It can be produced with TSH, it can also be produced with forskolin. If you prevent internalization of receptor, so you prevent the second wave of cyclic AMP that comes from the inside of a cell, you see with TSH no longer the remodeling of the actin cytoskeleton, you see no longer the appearance of phospho-VASP. It's not because the cell can't do it, because to forskolin, the direct stimulator of cyclic AMP production, it still reacts. So the cell can distinguish between cyclic AMP produced at the cell surface and cyclic AMP produced in the second wave in the intracellular space. So these two pools of cyclic AMP appear to be different. It's an interesting and difficult question how a cell can actually know where the cyclic AMP is being produced. And I'll show you at the end now just a single experiment that may illustrate how much a cell can compartmentalize or restrict the diffusion of cyclic AMP. These are experiments that are being done by Isabella Maiellaro in the lab, where she looks at a nerve. So this is a motor nerve in a fly, the nerve innervating a muscle, and she stimulates very locally a single bouton in these nerve cells. And she looks at the cyclic AMP in these cells when she does not stimulate or when she stimulates the receptors at the surface of the cell. And to facilitate viewing this, here is a film that shows just the differences that occur. So cyclic AMP going up is in red and cyclic AMP going down is in blue. And it's in response to a local stimulus that's delivered here to the receptors on a single bouton. And what you'll see here is a change that occurs very, very slowly and very localized. So the cyclic AMP will begin to appear here. You see this in red. It stays in a single bouton, just a little impulse on the neighboring boutons, and then it goes down, and these changes in blue are also limited to the single bouton, which means cyclic AMP, although you think it's a diffusible messenger, seems to be constrained in a cell and can be constrained to a very small volume of about 1 micron or so. How the cell does it is completely unknown. So this might allow us to distinguish between responses that are produced by receptors in different localizations and might allow us to develop drugs which produce only the first wave of receptor stimulation at the cell surface and has a set of physiological responses, or the second wave of signaling that occurs intracellularly and produces other types of responses. Now, in my last slide, I want to mention the fact that to search for compounds that have these distinct effects there's been tremendous progress in what we call virtual screening. It's a technology that I thought would not work at all a few years ago, but particularly Brian Scheuket at UCSF has shown that it's now actually possible to use a receptor structure and to dock in the computer, to dock every available chemical to this structure and select on the computer compounds that might be interesting. In doing so, they've increased the hit rate, which in pharmaceutical industry screening is usually 1 in a million or so, to about 1 in 5, which means you screen the compounds on the computer and then you select those that a computer tells you are interesting and actually test only those. If this holds true for more receptors than have been tested so far, it would allow us to really dip into a virtual unlimited number of compounds that we can study that will be producing interesting effects, stimulating only part of the whole plethora of responses that a receptor can cause. Let me summarize a few points. First, GPCR activation begins with very subtle changes, which are often not enough to develop interesting drugs by just targeting the orthosteric site of a receptor. Second, the receptors are floppy, which makes it difficult for them to transduce a signal, but it allows them also to adopt multiple conformations that could produce distinct downstream signals. And we may be able to stabilize these distinct conformations with ligands that bind to diverse binding sites that can be found in individual receptors, maybe alone, maybe in combination of two drugs that occupy different sites in the same receptor. Finally, receptor signaling appears to occur in distinct waves of signal that are different in their biochemical nature, different in their spatial nature, and different in their temporal patterns. And they may be producing distinct physiological downstream responses, and by utilizing these different waves, we can perhaps produce responses that are only part of what we see with conventional drugs. And these new types of drugs will hopefully be found not only by conventional screening, which is cumbersome, but also by virtual screening. And with this, I would like to thank you for your attention and would like to thank the people who have done this work and helped. Thank you.